Ever just stop and think about like all this stuff happening inside a single tiny microbial cell?
Yeah,
They're constantly juggling resources and reacting to everything going on around them.
right.
It's like some super intricate tiny machine just chucking away.
Absolutely incredible.
So today we're diving into how they manage all of that.
Yeah,
It's like looking at the operating system of a microbe,
exactly. The regulatory systems that control which genes get turned on or off.
and then how those proteins actually function.
Yep.
So we've got this really detailed overview. It covers everything from the proteins that interact with DNA to RNA molecules and even how the proteins themselves are controlled.
All the key players.
Our goal is to give you a good grasp of this amazing world of microbial regulation.
By the end, you should have a clear picture of all the different strategies microbes use to manage their internal processes.
So they can survive and adapt to their environment.
And thrive. It's all about efficiency.
So where do we even start? Our source material tells us that a lot of control in prokaryotes especially happens at the very beginning, with transcription.
Right at the start.
And that's mainly driven by regulatory proteins that bind to DNA.
Yep, that's a foundational concept. Our chapter highlights that some proteins and RNAs are always there, produced constitutively, like essential parts of the machinery.
Right, like always on.
But others are only made when they're needed, under very specific conditions.
Okay, so like on-demand production.
Exactly. That's key for efficiency. No point wasting energy making something you don't need, right?
Makes sense. So these DNA binding proteins, they're not just randomly attaching, are they? They have specific shapes.
Definitely not random. They have specific shapes that allow them to recognize certain DNA sequences.
The chapter mentions this helix-turn-helix motif.
Ah, yeah, like a key fitting into a lock.
So it's like one part of the protein, the recognition helix, actually makes contact with the DNA sequence.
Makes that connection.
And the other helix helps to stabilize the whole structure.
Right, keeps it all together.
This helix-turn-helix is super common in bacterial repressors and even some viruses.
Yeah, it's super common. You see it everywhere.
So they're not just blindly grabbing on, they're interacting with the chemical structure of the DNA itself.
Right, those nitrogenous bases, the backbone.
And often these proteins work in pairs,
Like a dynamic duo.
homodimers, and they bind to these mirrored DNA sequences.
Inverted repeats.
Yeah, so it's like they need a partner to hold on tight.
And they're looking for that reflected signal on the DNA,
These interactions often happen in the major groove of the DNA helix.
where there's more information for the protein to read.
So these DNA binding proteins, they're also called transcription factors, right?
That's right, yeah.
And they can basically do two things. Switch genes on or off.
Hit the gas or slam on the brakes.
So we have activator proteins that help RNA polymerase get to the promoter and start transcription.
Like pushing that accelerator pedal.
And the repressor proteins that bind to the operator and block RNA polymerase.
Hitting the brake.
But here's the thing. The activity of these transcription factors, it's not always the same.
It can change, for sure.
It's often controlled by these small molecules called effector molecules,
It's signals, basically.
like tiny switches. The chapter mentions inducers.
Inducers can kick things off,
They can trigger transcription by binding to an activator and making it more effective,
making it work better.
or by binding to a repressor and basically stopping it from binding to the DNA.
Yeah, like disabling the brakes.
And then there are core pressors.
Core pressors. They help the repressor clamp down tighter on the DNA, really shut things down. It's all about managing resources effectively. The chapter gives a perfect example with enzyme repression.
Oh, right. So if the cell has already made enough of a product, that product can act as a core pressor itself.
Exactly. It binds to the repressor protein.
So the repressor blocks the genes that make the enzymes that make even more of that product.
The classic negative feedback loop.
Keeps things balanced.
And then on the other side, there's enzyme induction.
So if a microbe runs into a new food source, it can trigger the production of the enzymes it needs to break it down.
Responds to what's available.
The chapter also makes this distinction. When repressors are involved, it's called negative control.
Because the gene is usually on by default.
And the repressor has to actively step in to stop it.
Right. And then there's positive control, where you need an activator to bind to boost transcription.
Now let's talk about operons.
Operons, I love this stuff. It's such a cool concept.
It's like having a bunch of related genes all controlled by a single regulatory site.
So efficient. Common in bacteria and archaea.
So if you need several enzymes to break down a sugar, you can control them all as a single unit.
All responding to the same signals from the environment.
And the chapter highlights this cool idea where a single small molecule can act like a master switch.
Like flipping a switch, turning a whole pathway on or off.
Now what about archaea? They don't have classic operons, do they?
No, they don't, but they still have activators and repressors.
So how do they manage coordinated gene regulation?
Well, instead of blocking RNA polymerase directly at an operator, repressors in archaea often prevent the binding of essential initiation factors,
Like what?
like TBP and TFB. Those are crucial for RNA polymerase to bind to the promoter.
So it's a different way of shutting things down.
Right, a different point of intervention.
And some regulatory proteins in archaea can even have two roles.
They can be both repressors and activators depending on where they bind on the DNA.
And on what other molecules are present.
Exactly. The context matters a lot.
Like having a key that can lock and unlock different doors depending on how you use it.
It is kind of like that.
Now let's move on to sensing and signal transduction. Microbes aren't just sitting there, are they? They need to know what's going on around them.
They need to respond.
So how do they do that? How do they detect changes and then relay that information?
Well, our chapter introduces two component regulatory systems.
Like a tag team. We have a sensor kinase stuck in the membrane.
Right there in the membrane.
And a response regulator hanging out in the cytoplasm.
Ready to respond.
How do they work together?
It's like a relay race. The sensor kinase detects some environmental signal, maybe a change in food availability or a chemical signal.
Like a warning signal.
Exactly. And then it adds a phosphate group to itself.
Phosphorylates itself.
Yep, autophosphorylation. And then it passes that phosphate to the response regulator.
As the baton.
Exactly. And that often changes the activity of the response regulator.
So what does it do?
Well, the response regulator is often a DNA binding protein, so it might bind to DNA and activate certain genes, or it could become a repressor and switch other genes off.
Two-way control.
But, of course, you need a way to reset the system, right?
Turn it off.
That's where phosphatases come in. They remove the phosphate group from the response regulator.
Back to its inactive state.
Like a reset button.
The chapter gives a couple of examples in E. coli, like the NVZ-OMPR system.
That one helps the bacteria deal with changes in osmolarity, basically how salty the environment is.
By controlling those porrin proteins, right?
The channels that let things in and out of the cell.
So depending on how salty it is, they can adjust the size and number of those channels.
Exactly. And then there's the NTR system, super important for managing nitrogen.
Which makes sense. Nitrogen is essential for building all sorts of stuff.
Absolutely. Proteins, nucleic acids.
The NTR system makes sure the cell has the right enzymes to scavenge nitrogen.
Based on what's available.
Okay, moving on from two-component systems, let's talk about chemotaxis.
Chemotaxis. This is where it gets really cool.
This is how bacteria move toward attractants and away from repellents, right?
They're not just floating around aimlessly. It's like they have a sense of smell or taste.
But instead of a nose or a tongue.
They have these methyl-accepting chemotaxis proteins
MCPs.
grouped together in chemoreceptor arrays.
Very organized.
And one crucial point, bacteria sense temporal gradients.
Temporal gradients. So they're not comparing the concentration of the front versus the back of the cell.
No, they're monitoring how the concentration changes as they move.
So they're constantly taking these tiny samples as they swim.
Like little sniff tests.
And these MCPs, they interact with a sensor kinase called CHE.
Right, and a coupling protein called CHU.
Now, what happens when an MCP detects something good, an attractant?
It inhibits the activity of CHE.
So CHE can't phosphorylate itself.
Exactly. And if CHE isn't phosphorylated, it can't phosphorylate another protein called CHE.
This is getting complicated.
But stay with me. Phosphorylated CHE, or CHE-YP, is what causes the bacterial flagella to rotate clockwise.
Clockwise, and that means?
Tumbling, randomly changing direction.
Okay, so if CHE-YP levels are low, the bacteria swims smoothly forward.
Run towards the attractant.
So attractant means run, got it. What about repellents?
Well, if they encounter a repellent or if they move away from an attractant, the opposite happens.
CHE gets phosphorylated.
Yep. And it phosphorylates CHE, leading to more CHE-YP and more tumbling.
So they can reorient and hopefully find a more direction.
Exactly. And then there's this other protein, CHE, that acts like a reset button.
It constantly dephosphorylates CHE-Phi.
Yep. Keeps things in check.
And then there's adaptation.
Adaptation. This is where it gets even cooler.
So even if they're exposed to a constant level of an attractant, they eventually get used to it.
Right. They don't keep reacting forever.
How does that happen?
Through methylation and demethylation of those MCPs, enzymes called Sher and Chi are involved.
So they can basically adjust their sensitivity.
And stay responsive to further changes in concentration.
It's like they're making tiny course corrections based on their environment.
Exactly. They're not just passively drifting.
They're actively sensing, processing information, and moving with purpose.
It's amazing.
Now, shifting gears a bit, let's talk about cell-to-cell signaling.
Quorum sensing.
Bacteria talking to each other.
It's like they're having a conversation.
So how does that work?
They produce and detect these small signaling molecules called autoinducers.
It's like they're asking, are there enough of us around to do something together?
Exactly. And when the concentration of these autoinducers reaches a certain level,
They launch a coordinated attack.
well, not always an attack, but a coordinated response, like producing virulence factors,
Or forming a biofilm.
which can be pretty impressive feats of coordination.
Gram-negative bacteria often use molecules like AHLs or AI2,
And gram-positive bacteria tend to use small peptides.
different languages.
That's a great way to put it.
And we get some really cool examples in the chapter, like bioluminescence in Alvibrio fishery,
The classic one. They only light up when there are enough of them around.
controlled by an AHL autoinducer and the Luxar protein.
Right. And then there's Shiga toxin-producing E. coli. The production of those nasty toxins is triggered by an AI3 autoinducer.
And even signals from the host organism.
Hormones, for instance.
Wow, so it's like a conversation between the bacteria and their host.
Exactly. And then in Staphylococcus aureus, the AIP and the agar system control virulence factor production.
So many different ways to communicate.
It really challenges our view of bacteria as just solitary creatures.
They're social. They work together.
And that opens up all sorts of possibilities for understanding and even manipulating their behavior.
Especially in infectious diseases. Maybe we can disrupt their communication networks.
Potentially.
Now let's move on to global control systems.
Where one signal can trigger changes in lots of genes across the whole genome.
Like a system-wide response.
Exactly. Adapting to big changes in their surroundings.
The chapter starts with the classic lac operon.
Ah, the lac operon, everyone's favorite example of gene regulation,
So it's induced by lactose.
which gets converted to allolactose,
And allolactose binds to the LAC-I repressor.
preventing it from binding to the operator.
So the genes needed to break down, lactose can be transcribed.
Right. But there's another layer of control here.
Catabolite repression.
That ensures E. coli uses glucose first if it's available.
Make sense? Glucose is easier to use.
More efficient.
And this involves the CRP protein,
The cyclic AMP receptor protein.
which is an activator. But it needs to bind to CMP to work.
Cyclic AMP and glucose actually inhibits DMP synthesis.
So when glucose is high, CMP is low.
Exactly. CRP is inactive, and even if the LAC-I repressor isn't bound to the lac operon because lactose is there, you still don't get much transcription.
So you need both lactose present and glucose absent to get the lac operon going full steam.
Exactly. It's a dual sensing system.
Make sure they're using the best energy source first.
Efficiency is key,
And the chapter mentions dioxic growth.
where they use up all the glucose first and then switch to lactose.
2 phases of growth.
A clear sign of this preference for glucose.
Okay, next up are the stringent and general stress responses.
Emergency protocols. When things get tough,
The stringent response happens when the cell is running out of amino acids.
essential building blocks.
So it slows down the synthesis of ribosomes and transfer RNAs.
The machinery of protein production.
And it turns on pathways to make the missing amino acids.
And even stop cell division.
Conserve resources.
Exactly.
And this is all mediated by alarm mode.
PPGPP and PPPGPP.
Quite a mouthful.
They are. Primarily made by the RLA enzyme.
Which gets activated when ribosomes get stuck because they can't find the right tRNAs.
No amino acids, no tRNAs, RLA kicks in.
And then there's spot T, which can also make and break down these alarmones.
Fine-tuning the response.
The stringent response isn't just about slowing down, though, is it?
No, it's about survival.
It's crucial for bacteria to handle tough times.
And it's even linked to persister cell formation.
Those super-resistant cells,
Yep.
like in Mycobacterium tuberculosis.
Exactly.
Then there's the general stress response,
A broader response to all sorts of challenges.
often controlled by alternative sigma factors, like RPOS in E. coli.
Right. Instead of the usual sigma factor, RPOS helps RNA polymerase recognize a different set of promoters.
The ones that control genes for dealing with stress.
DNA repair, protection against reactive oxygen species.
And there's a connection between the RPOS regulon and the stringent response.
They're linked.
It's like all the emergency systems are connected.
Makes sense. Right.
So when times are tough, the cell goes into economy mode.
Conserving energy, prioritizing survival.
Next, we have the phosphate, or PHO, regulon.
That one deals with phosphate shortages.
Phosphate. Why is it so important?
Oh, it's crucial for all sorts of things. DNA, RNA, ATP.
Energy currency.
You got it.
So the phoregulon uses a two-component system.
Typically with a sensor kinase like PhoRR and a response regulator like PhoB or PhoB.
And when phosphate levels drop, the sensor kinase gets activated.
Phosphorylates the response regulator.
What happens then?
Well, the phosphorylated response regulator binds to these DNA sequences called boxes.
And that turns on genes for getting more phosphate.
Uptake from the environment, breaking down organic phosphates.
Even storing phosphate when it's available again.
Exactly. When you're running low, you got to find it wherever you can.
But the chapter also says that phosphorylated PhoP can also repress some genes.
That's right, specifically some involved in nitrogen metabolism.
Why would it do that?
It's about prioritizing. If you're really short on phosphate, you don't want to waste energy on other processes.
Even essential ones like nitrogen assimilation.
Is it a trade-off?
And the phoregulon can also be involved in things like pathogenesis and biofilm formation.
And even antimicrobial resistance and toxin production in some bacteria.
Wow. Phosphate shortage really triggers a lot of changes.
It's amazing how a single missing resource can have such a wide impact.
It's all interconnected. Now, the last global control system is the heat shock response.
Protecting the cell from high temperatures and other stressors.
When things get too hot,
Exactly. Proteins can start to unfold or clump together.
lose their shape and stop working.
And that's bad news for the cell.
So the cell makes more heat shock proteins,
HSP.
molecular chaperones,
Helping to refold those damaged proteins or get rid of them altogether.
like cellular repair specialists.
That's a great way to think about it.
The chapter mentions HSP 100, HSP 90, HSP 70,
DNAK and E. coli.
and HSP 60, like GROAL, and HSP 10, like GROAS.
A whole team of helpers.
And the expression of these HSP genes is often controlled by alternative sigma factors.
Like RPOH and E. coli.
And RPOH is regulated at multiple levels.
Transcription, translation, even post-translation.
What does that mean?
It means the cell has several ways to control RPOH activity. Under normal conditions, DEC actually targets RPOH for degradation.
Keeps its levels low.
But when the cell is stressed, DEAC is busy fixing other proteins.
So RPOH can stick around and do its job.
Exactly.
It's like the cell has its own emergency repair system.
Ready to deploy when things get too hot.
Okay, we've covered DNA-binding proteins and global control systems.
Now for the world of RNA-based regulation.
It's always amazing to see how many important roles these non-coding RNAs have.
They're not just junk DNA.
Definitely not. They play a huge part in controlling gene expression.
Both at the transcriptional and translational levels.
The chapter starts with small regulatory RNAs.
sRNAs.
Yeah.
Little bit powerful.
They're typically between 40 and 400 nucleotides long.
Tiny RNAs.
And they work by binding to other RNAs.
Usually mRNAs.
And this binding can block the ribosome binding site.
Inhibiting translation. No protein produced.
Or it can expose the ribosome binding site.
Making translation more efficient.
And it can also affect how stable the mRNA is.
Leading to its degradation or protection.
So much control from such a small molecule.
Right. And there are two main types of sRNAs.
Transacting and cisacting.
Transacting sRNAs are encoded somewhere else in the genome.
Away from their target mRNAs.
And they often need help from chaperone proteins like HFQ to bind properly.
And cisacting sRNAs are encoded on the opposite strand of their target mRNA.
So they can bind directly.
The chapter gives some cool examples of transacting sRNAs.
Like RIHB.
That one's produced when iron is scarce.
It helps to conserve iron by reducing the production of iron-containing proteins.
Why?
And then there's SGRS.
That one's produced during glucose phosphate stress.
It triggers the destruction of the PTSG mRNA, which is part of the glucose uptake system.
It's like they're acting as switches, turning things on or off depending on what the cell needs.
Precisely.
So it's not just proteins controlling gene expression,
RNA is in the game too.
adding another layer of complexity.
And fine-tuning.
Now let's talk about riboswitches.
Riboswitches. These are fascinating.
They're these special RNA domains that can directly bind to metabolites.
Small molecules.
And this binding causes a change in the RNA structure,
A shape-shifting RNA.
which can affect translation or even transcription.
That's right. Unlike sRNAs, where the interaction is based on base pairing,
Riboswitches bind directly to the metabolite,
at a specific region called the aptamer.
so this binding can block the ribosome binding site,
No translation.
or it can cause the formation of a terminator structure.
Stopping transcription prematurely.
And the chapter points out that many riboswitches control genes involved in making the very metabolite they bind to.
A feedback loop.
So the levels of a molecule can directly regulate its own production.
It's a very elegant system.
The mRNA can sense and respond directly to the levels of a specific metabolite.
allowing for rapid adjustments in gene expression.
Lastly, let's talk about attenuation.
Attenuation, a classic mechanism.
This is all about stopping mRNA synthesis prematurely.
Before the whole message is transcribed.
The chapter explains that it depends on these alternative stem loop structures in the mRNA leader sequence.
Stem loops. They're crucial here.
And the formation of these stem loops depends on how a ribosome is doing, translating a short leader peptide.
It's like the ribosome is sending a signal.
The classic example is the tryptophan, or trypoperon in E. coli.
The textbook example.
So if tryptophan is plentiful,
The ribosome translates the leader peptide quickly,
no problem.
and that leads to the formation of a terminator stem loop.
Transcription stops.
But if tryptophan is scarce,
The ribosome stalls,
waiting for a tryptophan carrying tRNA
and that promotes the formation of an anti-terminator stem loop.
keeps transcription going.
So the ribosome is like a sensor for tryptophan levels.
Amazing, right?
The chapter also mentions that attenuation doesn't happen in eukaryotes.
Right, because transcription and translation happen in different places.
Nucleus versus cytoplasm.
Exactly.
This intricate coupling of transcription and translation in bacteria is really remarkable.
It allows for very responsive control of gene expression.
Okay, we've covered transcriptional and RNA-based regulation. Now, on to the last part of the chapter,
Regulation of enzymes and proteins after they've been made.
post-translational regulation,
It's like fine-tuning the machinery.
making adjustments on the fly.
The chapter starts with feedback inhibition.
We talked about that a little bit earlier, right?
Right, where the end product of a pathway inhibits an earlier enzyme.
It's like telling the factory to slow down production.
We have enough.
The chapter reminds us that this often happens through allosteric regulation,
Where the end product binds to a special site on the enzyme.
not the active site.
Right, the allosteric site,
And that changes the enzyme shape.
making it less active.
And they're also isoenzymes,
Different enzymes that do the same job but are regulated differently.
allowing for more precise control.
Exactly. It's all about fine-tuning.
So the cell can quickly adjust its metabolic activity as needed.
Efficient and responsive.
And then the chapter discusses all sorts of other post-translational modifications.
Phosphorylation, methylation, adenylativity, uridyeclitephen.
So many ways to tweak a protein.
It's like a toolbox of modifications.
We've already seen phosphorylation in 2-component systems and chemotaxis.
It's a key signaling mechanism
And the chapter brings up the regulation of PII proteins in nitrogen metabolism.
where the addition or removal of a uridylyl group affects their activity.
Small changes, big effects.
Absolutely.
And there are also anti-sigma factors,
Which bind to sigma factors and prevent them from starting transcription.
another way to quickly shut things down.
The chapter gives examples like RSEA and SPOII, AB,
Controlling sigma factors like RPOE and Ceph,
important for stress responses and endospore formation.
and even simple protein interactions can affect activity.
No modifications needed.
So the cell has all these different ways to control its proteins,
Both by regulating gene expression.
and by tweaking the proteins that have already been made.
It's a complex and fascinating system.
That was quite a journey.
It was a deep dive into microbial regulation.
We've covered everything from DNA-binding proteins to post-translational modifications.
From tiny switches to global control systems.
It's amazing how interconnected it all is.
All working together to make sure the microbe can survive and thrive.
So listeners, by understanding these basic principles, you can really appreciate how adaptable and complex even the smallest organisms are.
They're not just simple cells.
Definitely not.
And this knowledge can help us understand all sorts of other biological processes.
From metabolism to disease.
It's a foundation for further exploration.
It really makes you wonder how these regulatory systems evolved.
And how they're adapted and expanded upon in more complex life forms.
Something to think about.
Absolutely.
Well, that concludes our deep dive into microbial regulatory systems, as presented in the chapter. We covered all the key points, and hopefully you now have a better understanding of this fascinating and important area of biology.
We've covered it all. Thanks for listening.
Until next time.